We spend a lot of time on this pod discussing the scalability of ATMPs, or advanced therapy medicine products. Regenerative medicines like cell and gene therapies comprise a large population of ATMP manufacturers. And the reasons we talk so much about their scalability are at least twofold. First of all, scaling up the manufacture of cell and gene therapies to the point that they're both produced in enough volume and also accessible enough to meet the therapeutic demand is an incredibly large and complex challenge. Secondly, emerging cell and gene therapy biotechs are approaching those volume and accessibility challenges in dozens of different ways, from decentralizing autologous production and administration to pursuing any number of one-to-many allogeneic approaches. Today on the Business of Biotech, we're digging into those challenges with a company that sits at the bleeding edge of solving them. My guest is Charles Calderaro III, and he serves as Global Head of Technical Operations at Kite Pharma. Charles, welcome to the show.
Hey, it's a pleasure to be here. Thank you, Matt.
Oh, it's our pleasure to have you and our honor to have you. Now, I know you go by around the office, you go by Chuck. Do you prefer Chuck or Charles?
Chuck is perfectly fine. I think most folks call me that, except my mom usually when I'm in trouble. So, but Chuck is perfectly fine.
All right, I'm going to go with this. Isn't the first time we've talked, so I'm going to be presumptuous and assume that I'm comfortable enough with you to call you Chuck.
All right.
So I want to start by getting to know you a bit better, and I'm going to kind of go back in time a little ways to do that. Because you've got an interesting background starting with academia. You studied electrical and nuclear engineering in your college days before serving in the Navy as a commissioned officer and nuclear engineer. Thank you, first of all, for your service.
Thank you.
So when you left the Navy, it appears as though, on paper anyway, you went all in on the life sciences. So tell us about that transition and why that choice back then.
Oh, wow. You are taking me back. I was fortunate enough to interview with maybe a dozen companies from different industries and sectors, from oil and gas, nuclear power to, consumable products and some life sciences companies. And I was really drawn to life sciences because what was important to me is I wanted to be technically challenged and constantly learning. And two, I wanted to make a difference. One of the things I really enjoyed about being in the Navy is we made a difference. And I thought the biggest way I could make a difference was to help people with their health and with their lives. Then also, I wanted to get into a sector of industry that I felt had a long runway. And when I saw the opportunities to constantly learn and improve our standard of care for whatever disease we have to be talking about or working on, I just felt that life sciences kind of hit those three things: make a difference for people, scientifically, technically challenging and stimulating, and a long-term runway. And it's served me well. And I think I've been able to serve patients well, and the folks I've had a chance to work with over the last 30 some odd years. It's been a fun ride, and it's not over yet.
Yeah. And it's been a fun ride, and with some pretty impressive companies. I mean, you've spent some time in big bio with roles at Genentech, Roche, BioMarin. Tell me just briefly about those roles and how they sort of helped shape your worldview and ultimately kind of played into the preparation of your role here at Kite.
Thank you. It's a great question. The roles were fabulous and the companies were terrific, but I had great mentors and leaders and role models along my journey that gave me the right guidance at the right time. And there were 3 or 4 critical moments where I actually wanted to do something that it turned out I didn't do because of the feedback from these people who I trust very much. But I'd say for these companies all gave me an opportunity to work in pretty much all the functions of technical operations. And I think a first key milestone was at Genentech when I got to be a site head at the time, the world's largest and most complicated and automated monoclonal antibody manufacturing facility. That was the Genentech Vacaville facility in Northern California. And that was a terrific experience, not only from a science and perspective, but also from a leadership perspective. And it was something I had worked 10 or 15 years towards. It's probably the second thing from a global perspective. I would say the integration with Roche really helped broaden career opportunities for us just day to day, because we went from being a predominantly U.S. centric company, that in Genentech, to now we're part of this global company with more and more manufacturing facilities that we had a chance to work with, and then eventually led to a 4-year expat assignment in Switzerland, where I had an international assignment, but I had a global job, which that didn't quite click for me until I actually started doing the job. And when you travel 40% of the time and you're living abroad, you really can let go of your American bias or the unconscious bias that we have. We Americans tend to be, very U.S. centric, and California even more so California centric. And we're an important country, but we're not the only country. And we have to meet the needs and seek to understand the diversity of the rest of the world around us. And I think I really got that out of the experience with Roche and being an expat for 4 years. That took me to South America, Central America, Asia. I've been to these areas before, but to be living and working in this environment really helped me broaden that perspective.
Mm-hmm.
And I'd say the learning how to run a very large global company at an executive level, which is something I got exposed to in Basel, was also really cool and very, I'd say, transformative from my thinking on how to run the business.
Yeah. You mentioned, I just want to ask a follow-up question on that. You mentioned your work in monoclonal antibodies with Roche early on. Now that you're in, concertedly in the cell and gene space, are there parallels that you're drawing around sort of the scale up, and we're going to get into that discussion, but the scale up and regulatory scene around this new, much newer approach or modality? Can you draw parallels from that old experience at Roche with things like monoclonal antibodies back when they were sort of on the bleeding edge?
Yeah. So when I joined Genentech back in early in 2002 timeframe, monoclonal antibodies, recombinant engineering was still in its nascency. It was still being developed. We were developing products that changed how medicine was taught in school and practiced in the hospitals, and we were innovating science and manufacturing techniques to produce products that had never been made before. Mm-hmm. And we, having gone through that experience being with an innovator that pioneered a space, and then 20 years later getting a chance to kind of do it again, it's kind of like you kind of know what the twists and turns are. The things are different. It's not a cookie cutter, but you're kind of mentally prepared for, okay, we need to do this, and this, and we need to engage the health authorities, and we need to build partners with certain suppliers and develop skills and capabilities in our people that they can't get anywhere else. Now you can study recombinant engineering, monoclonal antibody manufacturing in universities, right? 20 years ago, you couldn't. It didn't exist, and we're kind of there now. And we're actually starting to work with local community colleges to create cell therapy biotech programs to help train people, educate people so they can then join us and be a part of our organization. But I'd say that I think the engagement with the health authorities, knowing that a lot of the standards, if you will, that exist in the industry before monoclonal antibodies, and a lot of the standards and compliance expectations that exist before cell therapy, had to be challenged because if we would apply those same rules, if you will, to monoclonals back then or to cell therapy today, we couldn't be doing what we're currently doing. A classic example, maybe a detail, but the industry standards for the duration of a sterility test is 14 days. And in cell therapy, we're able to do that in 7 days. And because of this advancing technology in doing that particular test, but also the openness of the health authorities, the FDA, the EMA, that, we can shorten the sterility test. And 5 years ago, that would be unthinkable, both from a regulator perspective and from a technology perspective. So a lot of parallels. The science is different. It's related, but there's a lot of parallels in how to scale and grow the business when you're the pioneer / innovator in a particular space.
Yeah. Yeah, very good. So I want to talk about this. I want to start the conversation around scaling manufacturing and scaling accessibility with a high level, I guess, your high level perception on the problem and the challenge, as it were. So, Kite isn't the only company that's, as I said from the outside of the conversation, scrambling to figure out how to scale up cell and gene therapies and make them more accessible. Why is it so important? And what is the challenge exactly?
Sure. Well, the importance of scaling the tech ops organization in cell therapy, the manufacturing capability is a strategic advantage. Um, for us, it really is essential if one is going to compete in the CAR T space, let alone deliver product to patients. And why is that? And I say this having worked in pretty much every modality that we have in the pharma biotech business. What cell therapy is the epitome of personalized medicine. Now, we've used that phrase in industry for well over a decade and a half, but cell therapy is personalized medicine. We start with the patient's cells, and from the moment of apheresis to the moment of product release, we get all that done in 17 days in the United States, and we get it done right perfectly, 97% the first time through. So that's a level of speed and performance that is unrivaled in other types of modalities. The other thing that is interesting in cell therapy, because we're starting with the patient's own T cells, there is no inventory. So you can't rely on inventory to make up for mistakes. We can't rely on inventory to hedge us in case there's an upside in demand or disruption in manufacturing. All other modalities, if you will, in our business is more of the make to stock type of supply chain, where we are make to order, personalized medicine. So I'd say those things are very interesting. The other thing is, and it's really analogous to Thomas Edison in the late 1800s. He invented two things. He typically gets only credit for one. And the first thing he created was obviously the light bulb. But folks sometimes don't realize he also created the alternating current power distribution grid. And in the late 1800s, in Lower Manhattan, he connected 60 homes to the power grid, and they hit the switch and they had electric light. Mm-hmm. Well, for us, our light bulb is Yescarta. It's taking the patient's T cells into one of our manufacturing facilities and transducing it with a viral vector, which is basically a cruise missile with a payload of DNA to instruct the T cell to produce a chimeric antigen receptor with in our case, a CD19 affinity for CD19. Okay. But our distribution, our power grid is the connection between 220 apheresis treatment centers and the two manufacturing facilities, soon to be three, that we have supporting, the US, Europe, rest of the world markets that we have. And what's different in cell therapy, and it's an expectation driven from the health authorities, is the manufacturer is accountable from a GMP compliance perspective from the moment of apheresis to the moment of infusion. That's not true in the make to stock world. One, you don't have to get the starting material from a human being. And two, it just sits on the shelf. Once it leaves your facility and gets purchased by a hospital or a wholesaler, then we're kind of no longer accountable for its journey to the patient, if you will, right? And so this network that connects the patient to the apheresis process that brings the cells to our plant, allows us to process and bring it back to the patient, right? That's our distribution system to the power grid. Then our light bulb is the actual manufacturing, if you will. And along this journey that the cells take from the treatment centers to the manufacturing plant and the manufacturing plant back to the treatment center, we use these two terms, chain of custody and chain of identity. And that's to make sure that the patient gets back the patient's cells and only their cells, and that their cells don't accidentally go someplace else to another patient, because that could be lethal to the recipient if the wrong cells are delivered. We've never had that mistake. And so we have to execute rapidly. We have to execute with a high level of reliability and a high level of precision and accuracy every single time, patient by patient. And so that's what we're challenged with when we start to scale. For Kite, we started with a facility in Southern California, and that's still our main facility today in El Segundo, which is just outside of LA, and it's licensed for all products in all markets. Last summer, our second facility was licensed in Europe. It's our Amsterdam facility, just outside the city of Amsterdam, and that is picking up the volume for all of Europe and Middle East countries. And that saves us the burden of having to fly patient cells from Europe across the Atlantic to Southern California and then back again. Obviously, that adds cost and time, which delays the therapy being reintroduced back to the patient. Yeah. So we've been we've relied heavily, not heavily, but this has been our model, is having a global network with regional hubs. Our third facility, which is under construction in Maryland, will pick up U.S. volume via backup for Europe, and that will free up our California facility to take on more markets in Latin America and Asia Pacific. So we feel very comfortable with that scalability. The other thing that was important to us is viral vector manufacturing. So we have pilot plants for viral vector and clinical manufacturing for viral vector, phase 1, phase 2, and phase 3, and then a commercial facility which will come online early next year. And that gives us the vertical integration of vector manufacturing and cell therapy manufacturing. Most other cell therapy companies, from our assessment, rely on third parties. That could triple the cost per batch of viral vector and more than quadruple the time it takes to manufacture the vector. It's a lot quicker and cheaper to do it internally. And we also have a clinical cell therapy manufacturing facility. So we feel between the vertical integration of viral vector, the clinical manufacturing facility, and a commercial network of 3 large facilities that have capacity to expand, we feel that has helped us meet the need of the over 200 and growing number of apheresis treatment centers and the new markets in Asia and Latin America and the Middle East that we're starting to target. So we're very excited by the future and the infrastructure we put in place to help us meet the current demand and future demand.
Yeah, let me ask you a follow up on that, because it strikes me, that in the age of a pandemic, for instance, you are fundamentally somewhat insulated from at least starting material supply chain disruptions, because your starting materials coming out of the patient. How has the pandemic, how have supply chain disruptions affected the manufacturing side of the business? Is the manufacturing side of the business a little bit more insulated in the model that you work within, or have there been some disruptions?
Yeah, that's a great question. To say that we had no disruptions, the folks at Kite would say, "What are you, crazy?" They had to do a lot to overcome the challenges that we had internally. Externally, through the lens of the patient, there were no disruptions. And I really credit our global supply chain leader, Laura Alquist, who manages trains, planes, and automobiles, worked through the border closings, worked with local authorities and our legal team, public affairs, to make sure that folks understood what it was we were transporting. And as you can imagine, in the early days of the pandemic, when flights were constantly getting canceled, you just can't take our cell therapy, which is shipped at minus 250 degrees Celsius. You just can't put that on any plane in any airport. There are certain controls and systems that have to be in place, going back to that chain of custody, chain of identity. It's not like we can just switch from one airline to another. It's all part of our that grid, that network I described earlier. We had to work very hard to overcome the logistical challenges, particularly in some remote countries where the apheresis treatment centers are far away from a hub. So Paris is an example of a hub in France, and there may be another dozen treatment centers that are within an hour's drive of the Paris hub. And then the cells flow through Paris, and then they go to one of our manufacturing facilities. And we had to then work in country to make sure that there was freedom of movement, not only within the country, but across some of the European borders, which there's sometimes, EU is 26 countries, and sometimes it's one country, and sometimes during things like a pandemic, countries start doing things independent, and we have to manage and work through that. The other challenge we had, which we overcame to meet the needs of the patient, is just working with our suppliers. There's a supplier for the kind of tubing that we use in the manufacturing process, or the reagents, or the starting material. On the surface, it may just be a component that one would grab off the shelf, or a chemical, or a biological agent, but it's part of a health saving therapy. And making sure those companies were given the same courtesy or access or freedom to operate under the pandemic that we had. So if you're Kite and you're part of Gilead, it's pretty obvious that we're here to save patients. But if you're a raw material supplier to Kite, it's not that obvious. So we had to help pull them into our space. We also had to work with our employees. And they were, our frontline employees were still able to come to work as we navigated and tried to understand the disease that was out there, and how to protect our employees, protect the product, and realize that every time an employee stepped out of their house to come to a Kite manufacturing facility, we had to take care of them. So we had a lot of things to overcome: raw material supplies, logistics and distribution, and creating a safe environment for our employees, while the sands of the pandemic were constantly shifting and changing. And we got smarter as this thing moved along. But it was really, really tough in the first 6 or 8 months. And I am truly amazed with the manufacturing team and the sites and our supply chain team that we never missed an apheresis, and we never missed a patient delivery. And no patient went without treatment during the pandemic.
Yeah, very, very cool. So I want to get your perception on some of the other approaches that I mentioned from the outset of our conversation. So, as you know, I've had on this show a handful of executives from emerging cell therapy companies who are approaching autologous cell therapies with big ideas, around let's do this in clinical settings from start to finish overnight, right? Like let's handle that like transplants were handled, back when that became in a clean room in a hospital, when it was a pioneering idea. What's your take on the feasibility of approaches like that, and what's Kite's take?
Well, I think for me, okay, and I don't want to speak for all of Kite, but for me, I think when I think of the nascency of the science and the challenges that come along with manufacturing, we're a long way away from turning that over to the hospitals that are not trained in this type of scientific, technically innovative manufacturing. And the testing that has to go along in process and for product release is also, some of those tests and some of those assays, if you will, are pull-throughs from biotech. Some of them are brand new that we had to invent to support the technology and the science that we're using to develop the CAR T. So, is there an opportunity for that down the road? Hey, anything's possible. But I'd say that the manufacturing process, the robustness, the portability isn't quite, in my opinion, is not quite there yet.
Yeah.
And I believe that the autologous cell therapy is really, from a technology perspective, continues to advance across the various heme malignancies, particularly those with CD19, and we're having the opportunity to progress later this year into second line, early next year into second line. I think as we continue to do that, we'll be able to bring more cell therapy to more patients and make a difference, and with very reliable, robust, and scalable manufacturing with a high level of precision and control over that process. And I think when you start dispersing the manufacturing process, that precision and the accuracy and would be compromised, in my opinion. At this point in the evolution and the nascency, if you will, of this technology.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. Yeah, and I can certainly appreciate those big ideas and the intent behind them, right? To sort of introduce new speed to the process, to improve accessibility, and I suppose arguably reduce costs. It occurs to me that when you transfer, when tech transfer happens from a manufacturing facility to potentially hundreds of clinics nationwide, that cost, the intended cost savings could certainly be offset. But I'm curious, from your perspective, from a tech ops perspective, in a manufacturing environment like that at Kite, where do you see opportunity to improve sort of the logistics efficiency and ultimately the cost of the therapy that's that patients and payers ultimately end up incurring?
Sure. No, it's a great question. Just building on something that you mentioned a moment ago, the logistics and distribution costs, are significant when you're flying cells across the Atlantic Ocean and back again. Mm-hmm. But when you're operating in region, in the European region, which is relatively small compared to the United States, and then you have a plant on the East Coast and a plant on the West Coast, those costs become more in line with that of a traditional pharmaceutical product, biotech product. Mm-hmm. Where we have the biggest cost challenge is from a cost of goods perspective. I don't think it's a secret to anyone that if you look at small molecules, aqueous drugs, biologics, biotech, vaccines, the cost of goods of a cell therapy product is pretty high. And it's largely right now because in the speed to bring this product into existence, there was a lot of manual steps along the way. They didn't slow down. We didn't slow down to build the automated perfect process. Process is very robust, but we didn't slow down to develop innovation from an automation perspective that hadn't been developed yet, right? Yeah. So we got to get the light bulb out there first before we start making a dimmer switch. And so for us, let's get Yescarta and Tecartus out there. Let's get the manufacturing process proven. And now we're working on, we started a partnership late 2017, 2016 timeframe with General Electric, which later became Cytiva, in automating the process that we currently have. And the TCF05, as we call it, T Cell Factory number 5, which is in the Frederick County, Maryland, that is designed and built around a process that's fully automated with the modules that we've co-developed with Cytiva. And that will basically reduce the labor to execute the process by about 50%. So that's probably the biggest innovation that we have coming from a process perspective. We're not innovating the process yet to the second generation process, but we are automating the process that we currently have with some enhancements along the way. There's also some opportunities to improve the assays that I mentioned earlier, the QC testing. I mentioned that there's some QC tests that we just pulled forward from biotech, but there's other ones that we had to create. And so now we want to be able to go back and say, how do we refine those, make them more reliable, and automate, and use more, leverage more automation, again, to reduce the help our workforce in QC become more efficient. Because unlike any other drug where you have a batch and you make one batch and you test one batch, and then you can treat 20,000 patients, we make one batch, we test one batch, and we treat one patient. Mm-hmm. So you're not going to be able to get around that, okay? But what we can do is automate and make more efficient the QC testing and the actual manufacturing to help reduce the labor force and get more. And we actually reduce the footprint or square footage per therapy that we have to manufacture in a plant. So those are the things that we're working on. We'll see that TCF05 Maryland facility. Then we have a plan to deploy the automation into the other facilities over a period of time.
Yeah. Well, thank you for entertaining those questions, Chuck. It's important that I ask you those questions because, recognizing that yes, Carta and Tecartus are pioneering therapies in this space, and Kite is a pioneer in the space. Early days, efficiency be damned, but now to get that perception right, insight from you into what's going on in terms of improving processes, improving efficiencies, so that we can make these therapies more accessible. I think it's an important response. And on that note, I mean, it's well known that yes, Carta and Tecartus are Kite's commercial products that are autologous therapies, but you're also working on allogeneic solutions as well. So I'm interested in where the manufacturing or technical challenges of that approach are kind of hitting Kite, and how you're addressing those challenges, and then maybe we'll talk a little bit about allogeneic versus autologous and where the space is headed from there.
Yeah. Well, I'd say that the innovation that we're pursuing in manufacturing and process development that's helping us advance our autologous programs will help us advance new technologies like allogenic therapy. From our perspective, allogenic therapy is not quite as ready for prime time as autologous. Mm-hmm. And we believe that even the first generation autologous therapies are unlikely to have the safety and potentially efficacy profile to treat patients in an out-of-hospital patient setting. So I think perhaps maybe the second generation of allogenic therapies may start to get to those level of safety and efficacy that we're starting to become comfortable and familiar with from a autologous therapy perspective. So for us, we continue to focus on the safety and efficacy for our patients today. And given the science and given the technology and the robustness and the information that we have today, we feel very confident in the continuing to pursue and refine the autologous therapy while we're still monitoring and developing our own allogenic programs.
Mm-hmm. Yeah, and I don't want to put you on the spot and ask you to pick a winner, but do you, if I did, would you kind of put your money on one or the other, or do you sort of think that moving forward, the industry as a whole, years down the road, that both approaches will move forward on their own merits?
That's a great question and a very fair question. But one of the things I've learned, having spent many years in the oncology space from a tech ops perspective, and I am not a clinician, but the probability that there's going to be a single solution for all cancers in all human beings is pretty low.
Yeah.
And what we see is combination therapies being efficacious. We see that some patients respond better to one type of therapy versus another type of therapy. There's no one magic bullet in oncology. And so the more different modalities, the more different treatments that we can get out there to improve the worldwide portfolio that doctors and clinicians can pull from to treat patients, I think in that scenario, everyone's a winner. And I think the focus is on the patients and delivering as many life-saving products and therapies as we possibly can. So, the short answer, yeah, there's going to be space for both, because I think patients are going to need one. Sometimes patients will need the other. And I think as long as we keep pursuing the science and sharing, we're going to help patients. That's the goal.
Yep. Okay. Now, you mentioned Cytiva, and I want you to vouch for me for a minute here, Chuck, before we move on. Vouch for me right now and tell our audience that when I approached you to be on this podcast, I had no idea that Kite was doing business with Cytiva. Cytiva is our sponsor, as you know. They're as you know now, they're the sponsor of this podcast. When I asked you to be on my podcast, I did not know that you were a Cytiva customer. So just let's get that out there.
No, absolutely. You did not know that. Actually, you were kind of your jaw dropped and you were shocked when at the same time when I did share that with you because of the reason you just mentioned. But no, ladies and gentlemen, there was no foreknowledge on Matt's part that Kite was working with Cytiva.
Thank you. I need to maintain my editorial credibility.
Yes, sir.
But you mentioned Cytiva's help as far as automation is concerned. So from a tech ops perspective, you're overseeing tech ops at Kite. If you stand back right now and you look at the manufacturing approach at Kite, and you recognize that you're taking steps toward efficiency via automation, where else, even theoretically, even if you don't have a solution in place right now, where else, when you look over sort of that manufacturing landscape, do you see opportunities or needs for further automation and efficiency?
Further automation or further innovation, or both?
Both. I would say automation and efficiency enabling innovation. So we know that this is a very expensive therapy. We know that the industry as a whole is striving to make it more cost efficient, make it more logistically efficient, and automation and innovation both play roles in those efforts. So I guess, again, I'm not putting you on the spot and asking you for solutions, but as you kind of survey that landscape, where do you see maybe the next big opportunities to solve a problem that will be, innovative opportunities to solve an efficiency enabling problem?
Yeah, no, a great question. And like I mentioned, we're currently automating the process that we have today, which brings one threshold, if you will, of efficiency. Mm-hmm. So then the next threshold would be, what's the next generation autologous cell therapy process look like? And on one hand, you can say, okay, how do we shorten the cell culture duration period, which is the longest step, where we actually grow and scale the cells and get them ready for dosing, if you will. So is there a way to shorten that process, have what we call a fast process, a fast CAR process? That's a combination of allowing some of the expansion to take place inside the patient, as well as being able to shorten the actual manufacturing process. So what does that look like? And once you get to this level of process improvement, it's kind of hard to do that without going back into the clinic. And that's one of the things in tech ops we always have to balance, is we want to make as much improvement as we can, but we don't necessarily want to go so far with the current process of a commercial product that it puts us back into a clinical trial to prove that we have comparability and that we have safety, et cetera, et cetera. So then for the next generation products coming through, we want to be able to say, okay, what can we do with the manufacturing process to make it shorter, and with a target around the cell culture step to begin with. The other thing that would be, which we don't have a solution for just yet, there's ideas and things that we are pursuing, but can you is there a way to transduce the T cell without using a viral vector, right? That viral vector, that's a whole nother manufacturing process that takes, a month plus to go prepare a batch of vector. And then that's the part where we bring the vector and put in contact with the T cell. Is there a way to use another type of technology, other type of method for transducing the T cell without using the vector, right? That eliminates another manufacturing step. That eliminates another cost. It means another thing that doesn't have to get managed from a supply chain perspective. So, and then QC, what else? Now that we've got more and more histories, there are ways in which we can work with the health authorities to make the testing, if you will, of the product less arduous, right? Because when you're doing something for the first time and you're not sure, the testing curriculum, if you will, could be pretty robust, and rightly so, right? But now that we have more experience, more safety data, more efficacy data, is there ways to maybe look at it differently? And so those are 4 areas where I see us focusing on moving forward in the autologous space. And Allogeneic, to your comment earlier, as it comes along also, in and of itself, as a make to stock, that starts to look and feel more like a traditional biotech product. And there's certain cost effectiveness and efficiencies in there, but then we still have to figure out how to make it. Even whether it's a healthy donor cell or an iPSC cell or an NK cell, you still got to transduce it. And you still got to take it through a cell therapy process where the culture has to grow. So you have the same challenges of process duration as well as transduction. The advantage is that you're not under pressure to turn it around in 17 days. You can make it, put it on the shelf, make it sit there for however long it's going to sit there. Yeah. The other challenge we have is with the temperatures that we move our product around. and this is more from a robustness and making it simpler perspective, you know, the temperatures that the cells come to us typically around 2 to 8 degrees, 2 to 15 degrees. And then when we send the product out, it's going out at minus 250 Celsius. And that's very expensive. That's arduous. And, you're dealing with liquid nitrogen, and having it, the product is stored in minus 250 liquid nitrogen. So is there other types of technology that we can do to create that type of cold environment beyond what we currently use today? Because that is also expensive and somewhat cumbersome. So those are 4 or 5 things that come to mind that I've learned. I've been at Kite for a year and a half. That's pretty much my cell therapy experience. I've learned a lot from my colleagues, and I'm looking forward to learning even more. But those would be the 5 things that I look at today and say, wow, if we can solve that for the next generation of products, we really advance the frontier of cell therapy more than we already have today.
Yeah. Well, we've got a pretty robust audience of new and emerging biotech executives with bright ideas. So maybe maybe those among our audience will grasp one of those 5 challenges and work with Kite to come up with a solution. Our new and emerging, the fact that our audience is new and emerging kind of leads me to my last tech ops question for you. And that's, given the experience that you've had with commercial stage companies all the way through your career, and now your experiences at Kite, which has experienced commercial success with CAR T therapies, what tech ops advice? And it is it's a terrible question. Don't tell me this is a good question. It's a terrible question because it's a great, big, giant, wide open question for you, Chuck. But what tech ops advice would you give that that leader of a new emerging cell therapy company?
That's a broad question.
Yeah, no, you can go ahead and use the word terrible.
No, it's not terrible. It's a great question. I'm very fortunate that I had the opportunity to join BioMarin and help start up its gene therapy program over a 3-year period in between the time that I repatriated with Genentech Roche and actually getting a chance to join Kite in my current role. And the learnings from that role were foundational and formidable to help prepare me for this role. First and foremost, in cell and gene therapy, because they're close cousins, process development, which is a very strong scientific component of tech ops, works closer with research and earlier in the life cycle of a program than in any other modality I've ever seen. So having a close relationship and aligned strategy with research is essential. And in other modalities, not so much, to the point that, our current head of research and we have joint offsites. Well, they're virtual right now, but we have joint working sessions. We go visit the research sites together with his leadership team and my scientific leaders to kind of get aligned so that we understand what they're trying to pursue, so we can create that scientific capability to get their ideas off the bench and into a IND, right? And with cell and gene therapy, the process is the product. And we say that a lot in biotech, but in cell and gene therapy, it's never more true, right? And so the close relationship with research is important. Second, the product is only as safe and as sound as we can characterize it. And that really means the rapid development in parallel of the assays, the analytical methods. Those sometimes get left behind a little bit. So I'm always very focused on analytical development because that enables QC testing. I spent a bunch of time talking about QC and robustness and reliability. That starts with analytical development. So we have analytical development, process development working side by side with our research colleagues, early research, this late stage research, preclinical, to make INDs happen. So I'd say that is a key area. And I was very fortunate while I was at BioMarin to really learn a lot about drug development in that gene therapy space, which was very transferable to Kite. The other thing I learned coming to Kite that I didn't know to expect is how closely tech ops works with commercial, right? In a traditional make to stock environment, you're working close with commercial when there's a stock out or there's a disruption in supply, and then someone from commercial calls the person in tech ops, says, "Where's my product?" And those apheresis treatment centers I talked about, that while our supply chain group and our quality group help manage those from a compliance and quality and supply chain perspective, but it's our marketing team, our commercial team, that's working with the clinicians, the oncologists, the patients, the practitioners, the hospitals, the insurance companies. So tech ops works side by side, commercial in the hospital at the treatment center with the patient. And that's so cool to see commercial and tech ops side by side. And when I joined Kite, the head of commercial said, tech ops is Kite's superpower. We can't do what we do for patients without you guys. And so those are two things in the cell and gene therapy space that I've learned, cell therapy space in particular, is the close relationship with research and obviously clinical development and with commercial. And that was probably the wow, I didn't realize that when I joined Kite. And so really for a tech ops leader, we talk about having an end-to-end enterprise perspective on things. Never been more important in this type of work environment to have, and also working closely with our business development colleagues, because a lot of the science and innovation doesn't come from one company; it usually comes from partnerships. So, on our Kite leadership team, we work very, very closely together. So, highly collaborative, bringing in right people that can think broadly across the organization. Strong scientific background, don't get me wrong, but these other little things are super, super important in the cell therapy space. Close work with research, close work with commercial, and always maintaining an enterprise perspective and engaging in ideas and in partnerships through our BD colleagues.
Yeah, that's very good advice. And not only is it's great advice, but you covered the gamut. You spoke to the new and emerging folks on the R&D side who maybe don't have anyone from commercial to talk to just yet, all the way up through commercial stage companies. So thank you for your transparency there. So we're running long here, Chuck. So I want to start wrapping things up, but I want to give you an opportunity to give me some thoughts on the pipeline, next steps at Kite. What's kind of coming down the pike, and then we'll wrap things up from there.
Oh, sure, great question, and thank you for it. I'd say for us, our number one priority is being able to bring cell therapy to more patients around the world. And Japan, we have approval in Japan right now for Yescarta. We have a positive opinion from China. We're expecting approval any day. There's other markets that are excited for us to bring cell therapy and to help in the markets we're currently in, how to improve access and continue to expand the label and indications that our current products have. We recently were approved for follicular lymphoma in the U.S. earlier this year in the third line. We have a PDUFA date in the September-October timeframe for acute lymphoblastic leukemia, ALL. That'll be a line extension for our Tecartus product, which is currently approved for mantle cell lymphoma, MCL. So we're very excited about the opportunities to reach more patients, reach more markets, improve access to cell therapy through educating patients and practitioners. And we're excited about the possibility of getting second-line approval early next year for DLBCL patients with Yescarta. So that's kind of, first and foremost. The other, we're excited about pursuing other types of targets in the hemalignancy space. We recently had Kite 222, which targets the CLL1 antigen for acute myeloid leukemia, AML. So that IND was approved late last year. We're looking forward to our first patient in shortly over the summertime. We're also pursuing programs with Bicistronic using dual targeting CARs, where we can target more than one antigen. We're excited about the possibility of advancing another program into the clinic, as well as continue to evaluate allogeneic programs that we have in our late-stage research preclinical phase program. And we're very active in the business development. One of our internal goals is to be a partner of choice, where companies can come to us that may not have the infrastructure that we've been very fortunate to create with the support financially from Gilead to help us create the infrastructure I just described earlier. And other companies, there's over 120 companies out there with 300 plus clinical trials. And we've learned a few things and made some mistakes along the way. And we feel like we can help other companies get their products to market and develop strategic partnerships and relationships. So those are kind of the things that we're focused on. Our new head of research, our new head of development, is super excited about being able to expand cell therapy into other modalities and in a solid tumor. Okay, so what other types of targets can we go after? I mean, 80% of cancer is solid tumor, and that's elusive because the targets are typically not on the surface of the cells. So how do we leverage TCRs, if you will, or find other targets that we can access? And we have a few in mind that our folks are looking at. But yeah, we're excited about that. So we're leveraging our strengths and broadening at the same time, and seeking partnerships to bring cell therapy to as many patients as we possibly can.
Well, it sounds as though you've got plenty of work to do. I mean, it's a very agree. What's next at Kite? Well, what's not next at Kite? You guys are working on a whole lot of stuff, and I applaud that work. You're obviously doing very important work. And I, we're the entire industry is thankful for it. And I want to mention this. Your leaders. I mean, it goes without saying that Kite's been as I said, a pioneer and a leader in this space. And I want you to know that I really appreciate the fact that you're willing to put that leadership on display by coming on this show and entertaining my questions and being as transparent with your responses and insightful with your responses as you've been. So thank you. Thank you for that. I really appreciate it.
Welcome. Pleasure to be here. I'm very humbled by the opportunity and appreciate the experience. Thank you.
Yeah, well, we're honored to have you, and I thank you. And we'll be paying attention and hopefully have you back on the show again when we've got some more to talk about.
Okay, thanks.
Thanks, Chuck. So that's Kite's Charles Calderaro III. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, which offers a trove of resources and guidance for emerging biopharmas at cytivalifesciences.com/emerging biotech. Check that out. Check us out at bioprocessonline.com, and please subscribe to my newsletter while you're there. In the meantime, before you go, give us 5 stars if you like what you heard, and thanks for listening.